Before the novel coronavirus became a full-blown global pandemic, there was a worldwide scramble to understand how and where it was spreading and who was getting sick. Critical information needed to guide public health decisions and save lives.
Today we're going to hear about a team of researchers in the Pacific Northwest whose work on another virus gave them a head start on SARS-CoV-2 and helped them find what, at the time, were the earliest known cases of community spread. Their work has gone on to provide invaluable data that social distancing works to slow down deadly viruses. I'm Rachel Tompa.
I'm Rob Piercy. And this is Lab Notes.
Hey, Rob, do you remember the winter of 2019?
Yeah, what was that, like a decade ago? Honestly, with the pandemic, I have lost all sense of time, so maybe it was a century.
Yeah, something like that. So for our listeners, we're in Seattle, and in early 2019, Seattle had this series of absolutely bonkers snowstorms.
More than 20 inches in the month of February alone, and that is a lot for Seattle.
Snow flurries blanketed residential neighborhoods, transforming local landscapes into seas of white.
People make fun of us Seattleites for our wimpy approach to snow. It's somewhat true, but it's also difficult to get around this city when it snows. There are a lot of hills around here.
The one county official called this storm a once-in-a-decade-or-two event.
Jokingly dubbed Snowpocalypse, many of us were stuck working from home, and schools shut down for several days.
The city working really hard to clear the streets.
At the time, I was like, when will my children ever get out of the house and go back to school? And I think that was like less than 2 weeks altogether. Can you imagine? Now it's been, also about a century since the kids have gone to school in person?
6 months.
Right, 6 months.
On Sunday, another storm could come through.
So that winter, we got this little taste of what it's like to be stuck in your house with no end in sight. But the snowpocalypse also hinted at the events of 2020 in another way.
And we've been collecting swabs, from all of the major hospitals throughout that period and testing them, not only for flu, but also for 25 other respiratory pathogens. And it was this really marked trend in the data where you could see that the fact of everyone staying home for a week or two really kind of seriously interrupted the transmission of many of these pathogens and the extent to which they were spreading.
That's Jay Shendure, a genomics researcher from the Allen Discovery Center at UW Medicine and scientific director of the Brotman Beatty Institute for Precision Medicine. He also leads the Seattle Flu Study, which has been kind of rebranded this year, as we'll get to later. The Seattle Flu Study was an epidemiological study to trace how influenza virus spreads in a large community, Seattle. The researchers would take nasal swabs from study volunteers and analyze the genome sequences of flu and other respiratory viruses in that swab sample. The researchers can figure out how viruses are transmitted based on their genetic sequence.
This is a concept you might have heard about recently. It's been done a lot with the novel coronavirus. But scientists have been using genetic sequences to trace the spread of other viruses for a while now. The flu study started a few months before the snowpocalypse, and they made this interesting observation. Let me just read you the title of the scientific article the flu study team wrote on the subject, because it's fascinating in light of where we are today. The title is Effects of Weather-Related Social Distancing on City-Scale Transmission of Respiratory Viruses.
Like Jay said, they got this first hint that staying in our houses, even just for a week or two, dramatically slowed the spread of flu and other respiratory viruses at a time that should have been the peak of flu season. This hadn't really been shown on such a large scale before. It was a weather-induced experiment in social distancing. That was the 2018-2019 flu season, and the study team felt well prepared coming into this season.
If we could go back to the beginning of the 2019-2020 flu season, how did things start out this year?
Yeah, so they started out great. I think we learned a lot from the first year of the flu study. It turned out it was good that we were prepared early and up and running because it wasn't early flu season. We were seeing a lot of flu cases early on. And I think largely the study was on track. And we were already starting to see, looking back at the year 1 data, even feeding into year 2, how the genome sequences of influenza could be leveraged to see patterns of transmission within Seattle. I think we were really digging into them at the time that things kind of took a turn.
When was it that there was this pivot, or when was the first red flag about this thing that was not flu?
I think that the moment that at least this really came onto my radar in kind of a in-your-face way was we had a PI meeting, and I think it was in early January, where Trevor Bedford, who's one of the other principal investigators of the flu study, brought this up as an agenda item, and that he'd been carefully reviewing all the available information. And I remember him saying, this could be 1918. It might not be, but it could be, and I'm worried. And he's the kind of person, I think, when he says something like that, you pay a lot of attention. And that really started us pivoting.
Trevor Bedford is a researcher at Fred Hutchinson Cancer Research Center, also here in Seattle. He's an expert on viral evolution and how viruses move around the world.
The 1918 flu pandemic he's talking about was the most severe pandemic in recent history, until this year.
These days, we're able to test for the genetic material in viruses in a widespread way. That's the basis for what the Seattle flu study was doing. They use a lab test called PCR to look for a piece of the flu genome and for other respiratory viruses.
After that meeting, the flu study team put together a PCR test for SARS-CoV-2, the novel coronavirus. They had that ready by late February. Any swab that came in as part of the flu study, they started testing for the coronavirus too.
Nothing on the first day, nothing on the second day. And then on the third day, we picked up a positive.
Did you guys think that it would be that fast that you get a positive result, like just on the third day after you started testing?
I don't think we knew, honestly. It was an honest-to-goodness research question. No one else was testing in the community, right? We had all these samples that were coming in, and we're in a position to test on them, and none of that testing was happening. We were genuinely surprised, I think. But at the same time, I think we wouldn't have been doing it if we didn't have some feeling that it might have been a possibility.
The first known positive case in the U.S. had been in mid-January near Seattle. There were no other positives for weeks after that, but nobody had really been looking for them. The testing criteria from the CDC at that time were really stringent.
Once they got that positive result of SARS-CoV-2 in the community, that set off a lot of other events.
I remember, I think I took 5 minutes, I think, to kind of absorb the information and then kind of kicked into gear. There's obviously a lot of things needed to get sorted out. We confirmed the result later that night. We sequenced the genome as quickly as we could, which Trevor then analyzed and suggested that community transmission had been going on for some time. I look back on my calendar for around then. I had a calendar item that says go to Costco, right? So, I think clearly at least some bug in my head was telling me that this could take a certain turn. Still, it was a big leap to imagine that things could go from where they were at that moment with really just a second case to how quickly they would spiral into what happened.
The Seattle flu study soon shifted its focus entirely to SARS-CoV-2. Jay and the other researchers partnered with the King County Public Health Department and relaunched as the Seattle Coronavirus Assessment Network, or SCAN, on March 23rd.
The SCAN program is a really simple but brilliant concept where people out in the community essentially become citizen scientists. How it works is you go to the SCAN website, enter your zip code, and if you meet all the requirements, they'll send you a test kit where you can collect a nasal swab at home.
I remember when the study launched, I read about it in the Seattle Times, and I was like, oh, this sounds cool. And I tried to sign up, and you couldn't. The site was just full right away.
Yeah, same. The first several times that I went to the website, the test kits were already gone by the time I got through. There was just a lot of interest out there in the community from people who wanted to be a part of this project. What happens, though, when you do get through is that a courier delivers a test kit right to your front doorstep. And then you have the distinct pleasure of sticking something up your nose for science.
Yeah, so let's start with the kit landing on your doorstep. You get it from your doorstep. There are pretty clear instructions in there. And, it's pretty simple. You're taking out a swab, which we provide. You're following instructions that are provided, and there are videos and other resources to make it as clear as possible for you to swab yourself or your child.
So I'm going to share my screen here. And my question is, am I doing this right?
Looks to me like you are. I think maybe you could go a little deeper, but I think you're doing pretty well.
Okay, so at this point in the interview, Rob was showing Jay a photo of himself taking the test. To describe this photo a little bit, Rob has his ring light and tripod set up on his dining table to get this beauty shot of him with a swab up his nose. And you're kind of wincing in the shot. It's great.
Yep. Spoiler alert, everyone. Rachel and I both got through on the website and got test kits, and then we documented the whole process.
So I just opened my package, and there's just a little blue box.
I'm actually pretty excited to do this. I've never been part of any kind of public health study or really any study, at least not that I know of.
It has a quick start guide, like it's an iPhone or something like that.
This is the test tube that you have to put the sample in.
Remove the swab from packaging.
This is the nasal swab right here.
All right, I need to blow my nose. I cannot believe I'm recording this.
All right, here we go.
Ooh, ow.
That's really unpleasant.
Okay, I had to pause because that was very uncomfortable.
Not quite touching the brain, but close. Just really want to sneeze right now.
Place the swab into the solution in the provided tube.
All right, and now we just seal it up.
That's it. Screw the cap on, and I'm done.
I am curious to know what the result is. So I send my test back, and I was actually surprised at just how quickly the courier came and picked it up. And within a day or two, I got the results back and negative.
Yay. Right. So yeah, I took the test too. I think I managed to get into the site maybe a week or two after you did. And I didn't quite have the same like citizen science excitement. It more reminded me of my grad school days, I guess, because I used to do a lot of PCR tests.
Yeah, important to point out for our listeners here, there's only one PhD biologist for whom this wasn't their first rodeo.
Yeah, that's me. I'm a former lab scientist, I suppose. But yeah, I stuck the swab all the way up my nose and mailed it off and then got the results back and womp womp.
Sad horns.
Inconclusive.
Which can happen with these sorts of tests if you don't have enough genetic material on the swab. For me, being a part of the SCAN project felt important. Like maybe I could make a difference in some small way and help scientists to learn something about this virus that could ultimately help them fight it. Have you gotten that sort of feedback from folks, Jay?
Yeah, we've definitely got that sentiment in a few different directions. One is just emails or things like that that are sent to the SCAN program and to the Seattle Flu Study. Another is sometimes we'll get a note clip, like a short written message or something like that comes back with the kit, and we'll often we'll just post those where people can see them. It is a real big team of people working hard here and putting in some real hours, including all the techs and the staff who really are critical for making this happen. And so, yeah, that feedback has been great.
So, in the 6 months since the study began, what have the SCAN researchers learned?
One of the big questions early on was whether there was some massive reservoir of cases in the community that was going completely undetected. Our testing, our early testing, so in those early months, I think, clearly painted a picture that was not the case. The overwhelming majority of individuals that we test who test positive either have some symptoms or, know someone who had symptoms, or are a household member of someone who, with someone who has symptoms or tested positive.
They're also measuring the prevalence of SARS-CoV-2, or the percent of people who are infected. And the results are matching what other groups tracking coronavirus prevalence in Seattle are seeing. All these different data streams eventually feed into the county's public health decisions around opening or closing parts of our community.
In the last few months, SCAN also started tracking behavior through simple questionnaires. in Washington state, we had a mask mandate for the general public put in place at the end of June.
Just in the last several weeks, as kind of the messaging has changed, we've definitely seen that mask usage has increased. But at the same time, we've also seen that social distancing has decreased. And in particular, that decreasing trend has been pronounced amongst individuals in their 20s and 30s, which is exactly the group that we're also seeing an increase in cases. I think testing is important, and I think testing is a critical part of all of this, is to understand what's going on and how things are getting transmitted through the community. But at the end of the day, the most important thing we can do right now is to really get people to adhere to the behaviors that are being recommended by public health. That's the most important thing.
This is just a general stumbling block about public health and population behavior, right? How do scientists and public health experts get people to adopt behaviors that are good for their health?
It might also help if we weren't all getting mixed messages on the subject.
I will say that I think that public health officials here in Washington and King County have been doing an outstanding job. The research community, the medical community, the testing community, a lot of things have come together to work. And so I think we're doing okay, relatively speaking, but obviously you can't let your eyes off the road. And I think maintaining that, it will be important to continue to keep doing all the things we're doing, as well as to keep paying attention to what public health is recommending and to follow that guidance very closely. Nationally, I think the picture is more dire, I think, with a lot of conflicting guidance coming from a lot of different levels. And we need a coherent national plan.
So where does Jay see us 6 months from now? A year from now?
So I think there's two very different futures 6 months from now. There's one future in which we have this under control. There's another scenario in which we're still struggling with this exactly the way that we are right now, with things bouncing back and forth in terms of opening and cases going up and down in different parts of the country. And so I think that's very much a choice, which of those roads we choose. And I'm hoping as soon as we can, we choose the road that leads us to that first possibility. In a year, I'm reasonably optimistic that some of these vaccines, for which there's encouraging data, are going to pan out. And so that's really what I'm hoping for. And, that a year from now, I'm getting my kids ready for school and, not having to think about a lot of the things that we're all thinking about right now with respect to whether that's going to happen or not.
Looking farther out, Jay hopes the lessons learned about social distancing during those snowbound days of 2019, as well as everything they have learned and will learn from the SCAN study, will influence better decisions down the road.
Then in 5 years, this is all memory, and our kids are called the Zoomer generation or something like that. And also things have flipped on how we view science and public health in terms of the trust being restored and if anything, enhanced. And that points us to a brighter future for the next time this happens, which it inevitably will.
I'm Rob Piercy.
I'm Rachel Tompa. For more Lab Notes episodes and science research news, visit our website at alleninstitute.org.
Thanks for listening.